you guys are going to you're going to use all the paper you want and all of the pretty colored pencils you want to draw out processes that work. You don't need them right now. You can get them right now if you want. Okay. So here's a cell. Okay? This cell is going to be a cell that's a very, very simple organism, or its genome is very simple. It's a cell where 2n equals 2. So what does that mean? How many pieces is the genome broken up into? 1. It isn't broken up into a bunch of pieces. The genome is intact in one piece. Okay? So there's, in this cell, there's going to be 2 double helices of DNA that are going to be the 2 homologous versions of the genome. One from mom, one from dad. So over here, I drew a DNA double helix in black pen and a DNA double helix in blue pen, and then the cartoon sketch version that we're going to use when I draw it up on the board. All right? So if I draw a black blob, you will interpret, I'm hoping, the black blob as a DNA double helix, a chromosome. Are we good with that? So the first thing we have to do when we're going to get ready to make a new cell, to go from 1 cell to 2 cells, is we have to copy the DNA. Right? So I'm going to go over here and I'm going to draw, get my little friend out of the way here. I'll draw my 2 chromosomes up here. First thing I have to do is I have to make a copy of each of these chromosomes. And so what that means, if you think back to the double helix, is I'm going to take each of these double helices and copy their information by separating them into their single strands of DNA and synthesizing a complementary sequence. So I'll synthesize a complementary sequence here, a complementary sequence here. I'll synthesize a complementary sequence of this one and do the same for the final one. So after I've done this, how similar are these 2 double helices? They're identical, right? They're 100% identical to each other, barring any mutation that happens to have happened. So these over here, what about them? Identical as well. So we have a name for these identical copies of chromosomes, and we call them sister chromatids. So this is a pair of sister chromatids. This is a pair of sister chromatids here as well. Now, those sister chromatids, after they're synthesized, often associate with each other, and they will associate with each other at that location called the centromere. So often you'll see these drawn out looking like this. We're not going to use this drawing scheme because I think it confuses people. But if you ever see it, just remember it's the 2 identical double helices that are sister chromatids associated with each other at that location called the centromere. So it's like exactly the same sequence on each one of these and exactly the same sequence on each one of these. Okay? So what I'll do over here is, first thing we always have to do with cell division is we copy the DNA. And in the process of cell division, we designate the copy phase with the letter S. And I don't tend to use these designations much myself, but the lab does occasionally refer to them. And so if it says draw chromosomes before or after the S phase, all it's referring to is before or after the copying of the DNA phase. So after copying, this would look like that. So pairs of sister chromatids, homologous chromosomes. Yeah.
So a chromosome, that X-shaped figure, which I know is complicated because of that, but like that is two sister pairs of
Yes.
of DNA together.
Yeah.
What,
Sister chromatids.
what do they compare to? If each one has the
They associate via proteins that basically adhere them together at the centromere.
Why? Oh, because of this process, right?
Well, one of the reasons, I don't know how deeply I can answer that question, why.
Okay.
But one reason it's important is we need them to stay together because at the end of the show, we want to end up with two cells that each have one black and one blue chromosome.
Yeah.
And so by keeping the two black ones together and the two blue ones together, you can sort of keep accounting of them better. I don't know if that answers your question. It's probably not as deep
the
as-
first part of an upper level part of it, yes.
Okay. So this, when you see chromosomes become visible in the cell, you've entered into this phase of mitosis. So from this point on, it's going to be all mitosis. And the very first phase of mitosis, I personally, some students really like to have words to associate with images so they can remember what to draw. I think it's a little bit misleading to talk about these phases because, as you saw in the video, they all flow into each other. It's not like a step and then another step and another step. There's a bunch of things that all happen simultaneously in this continuous motion. But when you see the chromosomes become visible, we call this prophase. And remember, what's the goal of mitosis? Separate the copies, right? So what we're going to do first is we're going to take our pairs of sister chromatids and we're going to line them up in the center of the cell. So if I redraw this cell and include an imaginary line down the center of the cell, well, it's actually not imaginary because I drew it, but in a cell it would be imaginary. I'm going to position these sister chromatid pairs down that center line. So I'll put my black chromosome here. I'll put the blue one down here. This is called metaphase. And they line up in metaphase.